Very good morning to everyone. In the training session on medicinal plant, today we will discuss about antimicrobial activity. So, for antimicrobial activity, first we need to identify and collect the experimental plant. So you can observe here that we collected the plant Abrus precarious. Here you can observe. Then we dried them. We made the powder. And after the powder, okay, we selected the solvent like N, hexane, methanol, aqueous, and acetone. So you can observe. Then we started the Soxlate. First powder material is weighed by using weighing machine, poured into the thimble which is made up of thick filter paper. Then the thimble is loaded into the main chamber of Soxlate extractor. The Soxlate extractor is placed into a flask containing the extraction solvent. Then Soxlate is occupied with a condenser. So this is the Soxlate apparatus, and you can observe. Then we started the antimicrobial activity. So for the antimicrobial activity, first preparation of working sealant using the agar media. So you can observe agar media, then autoclave, then oven dry for 1 to 2 hours, then pod and slant preparation, then streaking, then incubate for 24 hours. Then preparation of bacterial broth, nutrient broth preparation, then autoclave, oven dry for 1 to 2 hours, then poured into test tube, then inoculation of bacteria, then incubate for 24 hours. Now ready for antimicrobial activity test. So first is the dish diffusion assay. You can observe this is the dish diffusion assay. So complete a step, okay, you can observe and you can do. After this is the agar well diffusion assay. Okay, these are the steps. You can observe the zone of inhibition after agar well diffusion assay. Here you can observe the broad dilution assay for measuring minimum inhibitory concentration of a drug. So everything I illustrated here you can observe. This is the zone of diameters for the determination of successfulity with the standard dish diffusion method. So you can observe. So for the different types of antibiotics growth process. This is the before growth. Okay, the disc are placed antibiotic disc, then after growth you can observe if disc has a potential, you can observe a clear zone, no bacterial growth. That is called zone of inhibition. So zone of inhibition, if the inoculum is too light, the inhibition zone will be larger, although the sensitivity of the organism is unchanged. Relatively resistant strains may then be reported as susceptible. If the inoculum is too heavy, no inhibition zone size will be reduced and the susceptible strains may be reported as resistance. You can observe your like types of zone of inhibitions. So you can observe this is the this properly applied and this properly not applied. So here you can learn and you can do the antimicrobial activity. So possible error like inoculum too light, antibiotics too potent, QC strain muted, agar too thin, incorrect recording of result. Do not place the disc closer than 24 mm from center to center. No more than 12 discs should be placed on 150 mm plates or more than 5 discs on 100 mm plate. So measurement of zone of inhibition you can observe here. If growth occurs outside the ring, the organism is sensitive to antibiotics. If growth occurs within this ring, the organism is resistant to the antibiotics. If growth occurs within this ring, the organism has intermediate resistance or sensitivity. You can observe how to measure that we are measuring. Okay, so these are the way to measure the zone of inhibition. Okay, agar well diffusion you can observe. So these are the some zone of inhibition you can observe. We did lot of work. MIC result analysis you can observe here. Gradually decrease the in turbidity indicate inhibition of bacterial growth. MIC is the starting point of inhibition. Our experiment we did antibacterial activity of Abrus precarious against Salmonella typhi. You can observe again we start the work from field work. Then ethnobotanical information, collection of seeds, collection of plant specimen, herbarium deposition, voucher number, powder of collected seeds, extraction, crude extract from different solvent, then antimicrobial activities. So we selected three methods: agarwell diffusion assay, dish diffusion assay, and broth dilution assay. You can observe our work, the result. Always we, okay, do the zone of inhibition in mean. Okay, so you can observe. Here you can observe in this diffusion assay. This is the in broth dilution assay for estimation of minimum inhibitory concentration. You can observe here.